Good evening. My name's Loren Skow. I'm a senior professor in the Department of Veterinary Integrative Biosciences in the College of Veterinary Medicine. And on behalf of the organizing committee for this James Womack Memorial Symposium, welcome. This symposium seeks to honor the legacy of Jim Womack and his passion for studies of the comparative genomics of mammalian species. And the speakers in this symposium all shared a collegial and personal connection with Jim Womack. Some of them were his students, and many were his colleagues. And all of them enjoyed the hospitality of Jim and Raby Womack. And so if you haven't met the Womack family, they have joined us this evening. Raby and Jim's brother Gary and his wife Roseanne is somewhere. So we're glad you're here. This symposium could not have happened without the generous support of various units of the Texas A&M University, and I want to acknowledge that support. First and foremost, the College of Veterinary Medicine and Biomedical Sciences was very generous in supporting this symposium. Dr. August, Dr. Vimlapalli, and each of the 5 department heads made significant financial contributions. Dr. Criscitello is an Associate Dean for Research and Graduate Studies. He volunteered his staff to help support the arrangements for this symposium. And I want to give a special shout out to Ashley Seabury and April Orcutt. And I don't know if they're here, they may still be out there. They did a great job. The Hagler Institute for Advanced Studies and Dr. John Junkins, which is right here, were also generous supporters of the symposium, and thank you. Likewise, the Faculty Affairs provided financial support, as well as AgriLife and Dr. Cliff Lamb. And I should also thank the staff of the Hotel Conference Center, specifically Paige Hook and Rachel Forrester, for helping to put this symposium together. At this time, Dr. John August, the Dean of the College of Veterinary Medicine and Biomedical Sciences, is going to make a few remarks. Dr. August is a native of Surrey, England. He received his pre-vet education at Eastbourne College in Sussex, and in 1973 he graduated with honors from the Royal Veterinary College at the University of London. As a member of the Royal College of Veterinary Surgeons, he joined the faculty at Texas A&M in 1986 as head of the Small Animal Medicine and Surgery Department, and since 2020 he has served as the Carl B. King Dean of Veterinary Medicine. So please help me welcome Dr. August.
So howdy, and it is my privilege to serve our College of Veterinary Medicine and Biomedical Sciences as its Dean. And to everybody in attendance this evening, thank you so much for participating in this very special event, with an extra thank you to all of you who've traveled great distances to be here, and an extra thank you to our speakers as well. Welcome to Texas A&M University, home to nearly 80,000 students, a world-class faculty, superb staff, and an institution with a strong commitment to be on the forefront of the discipline of comparative genomics. And I'll reiterate what Loren said. We are so privileged to welcome Raby Womack to the conference. And Raby, we are so glad you can join us this evening for this very special celebration of Jim's remarkable career. I'll just reiterate something that Loren said as well. Conferences in academic venues require very careful planning, and on behalf of our college, I would first like to express my very sincere appreciation to Loren Skow for proposing the concept of this event and for bringing together the very best minds in the discipline. To our present presenters, again, thank you. And I suspect that once Loren and his colleagues, or one of his colleagues, told you who we would be honoring at this conference, it was a no-brainer to say yes to the invitation. And lastly, again, the 1,001 plus details required to make this event run smoothly have been very capably handled by Dr. Ashley Seabury, Miss April Orcutt, and their team from our Office of Research and Graduate Studies. Hats off to every one of you. Although my own academic path did not cross with Jim Womack's very often, I always knew him to be a humble man. When you think of the ideal scientific mentor, someone who is genuinely interested in the long-term success and happiness of their protégés, who is ethical, inquisitive, and who genuinely cares, Jim quickly comes to mind. Little wonder then that he graduated more than 50 doctoral students in his illustrious career in comparative genomics and genetics, many of whom are with us this evening in appreciation, and who perpetuate those same positive attributes in their own successful careers as Jim's. Often it's one person who influences and completely changes the course of our career paths. By coincidence, Jim and I shared that same experience. It was our biology teacher. If it wasn't for a biology teacher challenging Jim as an award-winning college basketball player to complete a genetics course he wanted to take, when Jim's career goal to that point was to coach high school basketball and teach math and science courses, we would not be gathered here this evening. I've absolutely no doubt that many of Jim's undergraduate students would share similar stories of the profound influence he played in their eventual career choices. One obituary for Jim, written lovingly by several colleagues here this evening, described Jim as the Lone Star pioneer of genomics. Jim's influence extended far beyond the borders of our state, exemplified by the extraordinary list of very highly prestigious awards that he received over his career. But knowing Jim, I suspect that he would have been equally thrilled by receiving the Wolf Prize and by the establishment of the James Womack Endowed Fund in Animal Genomics at Texas A&M University, created in his honor by his grateful former graduate students. How lucky we were when Jim returned to Texas from the Jackson Laboratory in 1977 to accept a faculty position at our college. His friend, Stephen O'Brien, encouraged him to consider changing his focus of research from mouse genetics to the bovine genome in support of his state's extensive cattle industry and its health challenges. And how Jim would have enjoyed collaborating now with our team of researchers at our Veterinary Education Research and Outreach campus in Canyon, just south of Amarillo. If you stick a pin in Canyon and then make a circle with a radius of 100 miles, you will find 7 million cattle living there at any given time. Little wonder then that this region is an extraordinary resource for our investigators who focus on resistance factors associated with bovine respiratory disease. Jim ascertained from his work that certain individual cattle and cattle breeds inherited resistance to infectious diseases, enabling them to evoke stronger defense mechanisms against, for example, bovine respiratory disease. Jim observed that cattle had more genes related to immune function than many other mammalian species. His goal was to develop a DNA chip that could be used as a practical diagnostic tool to identify the susceptibility or resistance of individual animals to these infectious agents. So we're here this evening and tomorrow to celebrate the life and accomplishments of an exemplary scientist, author of more than 350 peer-reviewed publications, whose name is inextricably linked with the term genomics. I read that rumor has it that Jim was part of the conversation when that term was first coined. Allow me to quote verbatim here from an obituary soon after Jim's passing. Jim's scientific contributions spanned nearly 50 years from the earliest days of comparative mammalian genetics to the sequencing of the bovine genome. His seminal paper that launched the field of comparative agricultural genomics was published in the Journal of Heredity in 1986. These studies revealed the deep conservation of chromosome organization in mammals, opening the door to the explosion in comparative genomics in farm animals and other species. End of quote. Jim's worldwide reputation and scientific impact continue to influence our university's commitment to comparative genomics, with David Threadgill, Bill Murphy, Penny Riggs, and other leading scientists charting new paths for the discipline at Texas A&M. The recent investment and significant investment in a new Institute of Comparative Genomics under Bill Murphy's direction within AgriLife Research and in partnership with the university holds great promise for many future scientific breakthroughs of which Jim would be very proud. So as we listen to some intriguing presentations this evening and tomorrow that point to the future of comparative genomics, and as we enjoy colleagues in conversation this evening and tomorrow, let's salute the legacy of Dr. James E. Womack, the Lone Star pioneer of genomics, member of the National Academy of Sciences, the first director of the Bovine Genome Project, gifted college basketball player, Abilene Christian University Sports Hall of Fame inductee, expert fly fisherman, and a humble giant. Thank you.
Our keynote speaker for this evening is Dr. Harris Lewin, and I've asked Dr. Larry Schook to introduce Harris, and come up here. While he's making his way up here, I want to let you know that we have this room and the cash bar until 10 o'clock. So I don't know how long Harris's lecture is, but if we have some time afterward, you are more than welcome to remain here and socialize after the talk. So with that, I'll ask you to help me welcome Dr. Larry Schook.
Well, then let me first thank Loren and the organizing committee for this invitation to participate. And Dean August, you made some comments I think were right on target. And I'm one of those things, and Jim taught me what Steve O'Brien taught him. I was a med school mouse geneticist until I introduced myself to Harris Lewin. And from that moment on, I just looked at pigs as big mice. And so that's been my career, I was just looking at that, and I said, that can't be too hard, but, it took, 40 years to get there, but we're getting there. So, believe it or not, I was Harris's birthday present back, and we met that is Harris up there, by the way, in 1986. And that's when my journey began with Harris, and they're intrigued because of Harris, that's how I got introduced to Jim, and that journey of how he counseled and mentored me in my own career. And I think the comments were spot on. It's like he was gentle, we were just talking about, he always had that smile when I'd say something stupid like, what's heredity or something like that? I don't understand. You either inherit it or you don't inherit it. And he'd just smile, and you've got a lot to learn, son. But, you know, it's good. So let me just start by quickly going through that. I met Harris in 1986. As I said, was recruited in the biotech center at Illinois from Medical College of Virginia. I'd been at University of Michigan, and so it was all mouse. And they were interested in back the same thing. We need to look at how we can bring mouse genetics, disease resistance into the farm animal system. So it was very consistent to that where Jim started with. And so I also, the first time I met Jim was actually in 1989 in Harris, and this was at the Banbury Conference that Jim organized. And it was really that came out with this idea of really creating an animal genome project in agriculture. And so I was very fortunate to be invited to do and work with that at that meeting and talk about the pig. So here you have on the left, here was the status of the pig genome in 1986, or 1989. It was, I'd say, anyone here would say that was bleak. And because of that, Jim was very helpful in how do we get organized. And I think building community is a spirit of what I he left me with, is how do we build community? And Jim did that not only in the United States, but internationally. And so Harris and I started a series of what were called the Allerton conferences. And we did that so we would bring in every couple of years and we'd really look at, and that's where we met our colleagues from Europe to come in and really talk about what's going on and how is each group attacking. You know, PigMap had just started in Europe. The bovine community was coming together. So we began to have these kinds of conferences, and we were very fortunate, and our deans supported this. And then it really became where Jim's national leadership was really when we created the Alliance for Animal Genome Research in the United States. And he really, and we did this, and that was I think, to me, a brilliant comment from him was, it can't just be a bunch of university professors saying this is what we're going to do. It really had to be a tripartite of the government agencies, industry, and academics. And so this is what the animal genome research was. We looked at targets. We began to have workshops in this area. And we actually hired Kelly Eversole to really kind of be the person who was going to collect money from everybody and where we could have our meetings and continue to do that. And I was telling Richard when he came in today that our first project, and this is one where, again, because of the success of the bovine cattle project that Jim organized here, we took the same step and put together a white paper process, and Max Rothschild and John Beaver and Richard were really good about how we could begin to build on how it was being done for cattle, and we could do that in pigs. So that was very successful. And then with Harris, his leadership on campus in Urbana, we were able to get together some, what I call piggy bank money, where we were able to get some support to begin to build the physical maps that were going to be required for sequencing. I just want to go that we were very fortunate that we had colleagues, and John Beaver, who's now head of the cattle project at the University of Tennessee, but we also, many people here had community where we had workshops, and again, Harris and Jim were always there to help build that community spot. Now, before I get too serious about Harris, there's another side of Harris that many of you have not seen, and you're very fortunate you haven't seen it. But part of this is he has been here in LaGrange, here in Texas for a while, and this is how we built spirit in our community. Was we had a band. We got together every summer and really had a celebration of where we were going. So Harris and his son joined the group later on. So we really built family. But this is a historic moment for me, and I was just sharing this was at 22/20, 1995. That was when, on the last play game, Texas A&M beat Michigan. And Jim and I had a bet. And the bet was whoever lost had to sing the other school's fight song. So the last play of the game, by the way, so Jim would have had to sing Hail to the Victors, and I had to sing the Aggie War Hymn. That was it. No money. This is all about pride. And I have this here with the tours. So in 1996, in Tours, France, if you were there at ISAG, Jim had them. Yep, he came, and we thought he had forgotten all about it. And this was in a castle, and the knights came out and trumpets. And Jim had them, they came over, and they took me up to center stage. And in front of, I don't know, 1,000 people, I had to sing the Aggie War Hymn. And I will tell you, he wished he'd never asked me to sing it. I'll put it that way. So that's my, with Jim. And let me now turn to Harris. And I think everyone here is aware, and the program has a lot, but I think there's several areas that I think he's continued to distinguish himself. And as I said, I met Harris when he was a assistant professor, and soon became my boss, so there you go. And at the University of Illinois, and I know he went to Davis, and he's still continuing to do activities, but we worked together for almost 30 years. So that was, and we continue to still do that. And Gutzel Endowed Chair in Immunogenetics. He was the founding director of our, if you will, our comparative genome center, which got kept funding, so that was something. And then he was the founding director of our Institute for Genomic Biology. And it was because of that moment, when that created, that's how he was able to, I was recruited back. I had been for a decade at the University of Minnesota in the vet school there, and came back to really become a theme leader. And that was just when we began to start the genome sequencing of the pig genome. So he is the father of that effort for me. I think all of us are really excited about his efforts to fund the Earth Biogenome Project, which is continuing to go on. And I think, again, building on that mammalian species and evolution is going to be a critical point. And the international recognition, like Jim, he won the Wolf Prize in Agriculture, which is, as we know, and we have several other members here, the National Academy of Swedish Academy of Agriculture and National Academy of Sciences. So it's with my deep personal appreciation that I get to introduce Harris to come up here, but also I want to make sure that it's all because of Jim that I'm actually here. So thank you very much.
Well, it's hard to listen to that about myself, but thanks, Larry, and revenge is coming, so don't worry. He was actually kind. I thought he'd be. He had other pictures that he could have shown that probably would have been a little bit more embarrassing, but he didn't.
Loren saw him.
Yeah, Loren vetoed that one? All right. Well, I have one that's kind of on the borderline maybe, but again, Larry, thanks very much. Old friend, like Larry said, almost 40 years now. And thank you, Loren, and all the other members of the organizing committee and all of Jim's friends and colleagues and family here. And most of all, thanks. I'm so delighted to see Raby here and Jim's brother, who I met in 2023 at Jim's funeral, and Mary, and people I've known for almost half a century, and it's just a tremendous honor for me to be here. Emotional, actually, and as you all know, Jim was an exceptional scientist. He was an exceptional scientist who impacted the lives and the careers of his trainees. We're privileged to have many of them here today. And scientific collaborators, of which there are also many here today, including myself, and was mentioned by the Dean. Dean really set me up. Dean August, thank you very much for that. But as you mentioned, his influence in the field of comparative genomics was immeasurable, and his legacy was global, as well as local in his community. Now, capturing the scope and the depth of Jim's career in 40 minutes is a great challenge, which is one reason why many of you here will be given the opportunity to speak over the next day or so to tell your own stories. And I look forward to hearing all of those stories as well. I believe that Jim effectively conveyed the essence of his career in his autobiographical article, which I commissioned as the editor of Annual Reviews in Animal Biosciences. He did an autobiographical article in 2018 called Mapping Genes is Good for You, which draws on one of his favorite quotes from Frank Ruddle, actually, who is a person that Jim deeply admired. And it's true, mapping genes benefited Jim, and it continues to benefit all of us, his scientific disciples. And the shared passion that we all have and our immense respect for Jim as a scientist and as a person are the common threads that unite us for this symposium. Let me just move on here a little bit. Yep. So Jim, as you all know, was my friend, and he was a scientific collaborator of mine for over 30 years. He helped my career at critical times, and many of which I will elaborate upon in my talk today. And I think our relationship is captured in this rather unusual picture taken at Jim's ranch. I'd like you to notice the following: the shoes, the pants, the belt, the hands in the pockets. It's pretty odd, isn't it? Well, it turns out that Jim and I share the same birthday, March 30th, just 16 years apart. But despite being born worlds apart and growing up under completely different circumstances, we had a lot in common. We loved academic life, we loved cattle, we loved genetics, we loved sports, and we loved our families. And there was an unspoken connection between Jim and myself that is really hard to describe, but it was there, and it lasted, as I said, for more than 30 years. So I'm going to take you on this tour, if you will, of Jim's career and his contributions, which were briefly touched upon by the Dean and by Larry. Jim began his scientific career as a mouse geneticist, as we've heard, and as a grad student in the laboratory of Ralph Bogart at Oregon State University. His first paper, which is shown there, that you can find if you search PubMed, was in 1968, and the effects of irradiation on the selection response in mice. And that was 1968, and after that, as Raby can tell you, that's when Jim returned to Abilene Christian University, and Jim and family went back there, and Jim took some of what he had learned during his time there, and he began to work with, he spent a summer at the Jackson Lab. This was a dream of Jim, of Bogart's, to have Jim to go to the Jackson Lab in Maine. And Jim spent a summer at Jackson Lab doing mutagenesis studies and various things that Tom Roderick and others had asked him to work on. Jim, while there, discovered rat allozymes. Specifically, here's his first paper of a genetic marker that he discovered, which was genetic variation in an esterase, in an enzyme that he could just collect from animal blood, and then you can separate out these markers by the electrophoretic properties usually in starch gels. That's how it was done. Oh yeah, sure, it's not changed. My apologies, thanks. Okay, yeah, thanks, that's better. Yeah, no, wait. Oh, here we go, yeah. I got two things, yeah. It's gonna be complicated for an old man, I know, it's not too good. So, okay, so sorry about that. And so, thanks, Larry. And so Jim's, again, his first paper was this discovery of the effects of irradiation on selection and response in mice. Then he discovered allozymes. And the first paper where he was involved in mapping was this, looking at the linkage relationships of rat esterases. And that's the first example in 1973. And Jim then, he was working with Tom, and he was then at about that time introduced to people like Steve O'Brien in '73. And this work on the first mapping of the rat esterases really put him into the big leagues, right? Because through his work on mapping, he met people like Frank Ruddle and Steve O'Brien and Victor McCusick. And they were the ones that formed the first committee, a comparative mapping committee, that came out of these meetings of human geneticists at that time. And so there were these series of workshops on human gene mapping, the first of which was held in 1973. And Jim became an integral player because he was one of the first to do mapping. And as I'm going to show in a moment, he was one of the first to do comparative mapping using these esterases, that system that he had discovered. Now, just to be sure, while he was there, he formed a very close relationship with Tom Roderick. And it was Tom Roderick, who the dean mentioned, who actually suggested the term over beers in Bethesda. There was an article written about this, the term genomics. They were looking for a title for a new journal. And it was Tom who bloated out, well, why not genomics? And I guess over the beer, Steve and Jim and a few others that were there all agreed. And that's how the term genomics was born. So Jim was at the very birth of the field of genomics. And I suspect that Dr. O'Brien will be telling us more about this, having been there at the time. Yeah, I said you were there. Yeah, sorry. Did I not? Okay. And so here it is. Here's Jim's first paper. Jim, in 1975 and 76, returned to Bar Harbor for a 2-year stint because Tom Roderick took an appointment at the Atomic Energy Commission. And so Jim and Raby went back to run Tom's lab while Tom was off and doing his thing with the Atomic Energy Commission, because he was interested in effects of radiation on the genome and such. And so Jim offered to run his lab, and he brought with him his first PhD student, who was Loren Skow. Okay? So you were there at that time, so you very well know I'm telling the truth here. Let me see, this is Larry's height, not my height. I think I have to bring this down a little bit. Let's see. Okay. So Jim then published the first comparative map, linkage map of the esterase family, a locus that you can see extended more than 15 centimorgans in the rat and mouse genomes. And again, this is Jim's first hand in comparative mapping and set the stage really for the next 40 years of Jim's career. And it's important to note that biochemical polymorphisms really provided the first resource for comparative genomics. In fact, I even entered the field. Gus Cothran's here. People who are here are old enough to remember when the only genetic markers we had were blood groups and biochemical polymorphisms. And Jim came at it from the mouse side, and myself and several others, Leif worked on in this area as well, and Gus and people like Ernie Bailey and, of course, Clyde Sormont and others I'll mention in a few minutes. This was what we had as a tool to do mapping. And that period ended, and Raby and Jim returned, again, took a different path. And after Bar Harbor, they came, as the dean mentioned, they came here to Texas A&M University in 1977, where Jim began his career in the Department of Veterinary Pathobiology. And it was also a place where Joe Templeton, who is another beloved member of the faculty here, had an appointment. And I knew Joe because I worked on the bovine majoristic compatibility complex. And I worked with Joe before I actually worked with Jim. And I loved Joe. He was a wonderful man and an excellent scientist. And so I guess it was Joe who convinced Jim to come. And that started it all off here at Texas A&M. Now, over the next 8 years or so, a lot of genes were mapped by Jim, mouse genes and rat genes. Mostly, Loren worked on rabbits, right, at Bar Harbor? Yeah. And Jim, in his autobiography, said probably the most exciting, and I need to move it again, probably the most exciting discovery that he made in that time was the discovery of a mouse with a mutation that influenced the level of neuraminidase in peripheral blood. And what that did was it had pleiotropic effect. It had downstream effects on the processing of other enzymes. And this was very exciting because there were models, all kinds of storage diseases and such. And Jim, Jim says this was the most important contribution he made at the time. He had it published in Science, and it was great, but it did not provide him with a solid foundation for funding. I'll return to that in a moment. So, in this lower picture here, which has a younger and much more handsome Steve O'Brien and a very handsome Jim Womack, and an ageless person, Jenny Graves. She looks exactly the same. She's 80 years old, more than 80, and she looks exactly the same as this picture. I don't know how I got this picture. I don't know if Jenny sent it to me or Jim sent it to me, but I had it in my archives, and I love this picture because it's of Jim and Steve and Jenny at one of these early comparative mapping workshops, sitting down, rolling up their sleeves, and figuring things out. And if you look at the text, I had to find this. It was almost impossible to get this in this journal, Cytogenetics and Cell Genetics. There are no online versions of these journals anymore, but somebody scanned this, and I was at least able to get part of the text. And look at what it says there. It's the gestalt of comparative genomics. Here it is already in 1984, and Jim and Tom and Steve are saying comparative genomics of a mapping of mammalian species has important ramifications. Among them is extrapolating from experimental mammals to the human, particularly in search for animal models of human genetic diseases. Another is to further understanding of genome and chromosome evolution with an objective of insights to genome organization and gene regulation. You can take that, cut it out, and put it in any grant that you are submitting today to the NIH or the USDA. You can take that text. It's still applicable today. This is 1984. And it's all there, already there, and in the minds of these three founding scientists in the field. And so at that time in 84, Jim came to a fork in the road in his career, also mentioned by the dean. And he says it was probably the most significant fork in his career path since the undergraduate genetics course that he took since his undergraduate genetics course. And he was at the home of Steve O'Brien and Diane O'Brien in Bethesda. And they were having a conversation because Jim was just having a lot of trouble getting funding. So Steve was trying to figure out what species should I be mapping? Should I be doing dogs? Should I be doing cattle? And Steve turned off the stereo and he said, "Haas, you're in Texas, right? Texas is for cowboys and cattle, not dogs." That's my best O'Brien imitation. And after a couple of more cycles of Blue Eyes, and that's for him, but I think it was Willie Nelson. I think it was Blue Eyes Crying in the Rain, because Blue Eyes is sung by somebody else, but Willie sung Blue Eyes Crying in the Rain. So you'll have to correct me if I'm wrong. I think it was Willie Nelson. And he went to bed thinking about agricultural research for the first time since telling Dr. Bogart that he had little interest in cattle genetics. That's why he did mouse genetics. Whoops, sorry. And so that really got Jim thinking. And indeed, Jim took Steve's advice. Whoops. Okay, let's see. Yep. All right, yep. Jim took Steve's advice and went back and made a somatic cell hybrid panel. First cattle somatic cell hybrid panel consisting, I think it was like 30 or so cell lines. And he published first his preliminary maps in Steve's book, Genetic Maps in 1984. That's where I first saw a cattle map. It was in Steve's book, the 1984 book. And then, of course, Jim went on with Yvonne Moll to publish this seminal article that was talked about in 1986, Womack and Moll. And I think, you know, this really was the contribution that he became most widely known for, was developing the first synteny map of the cattle genome, which helped to anchor the linkage maps to their proper chromosomes. And it's really this paper, Womack and Moll, which launched the entire field of agricultural genomics, because there were no other maps, really, very few maps. You saw one marker, maybe a few linkage groups here and there, but this was it. This was the start. This paper mapped 28 loci and into 28 independent linkage groups, and brought the entire composite map of the bovine genome in 1984, the year I got my PhD at UC Davis, to 35 loci on 24 syntenic groups. So he didn't have a marker even for each of the chromosomes. He had less than one marker. And Steve wants me to go. Go to the next slide. Yeah, well, I wasn't done with, okay. Yeah, no, I was there. I'm still there. Steve, he can coach me, he's allowed if he likes it. But so that was amazing. And that is the time that I first met Jim. In 1986, he came to the University of Illinois to give a seminar. He was invited there by Bill Daniel, an old friend of his, to give a talk about his new cattle map. I met with Jim. I'd read his work. I was absolutely, really enthusiastic about it because I had been working on the cattle major histocompatibility complex. One gene, one locus. We had been mapping genes for resistance and susceptibility to infectious diseases in cattle. But John Shattuck, who was a former dean here, told me, when I was an assistant professor, he said, a mouse is not just a chromosome 17 with a tail. So I figured, well, I guess then a cow is not a chromosome 23 with an udder. Maybe we ought to look more broadly at the genome, because by then Morris Soler had published his seminal papers and said, well, if we're going to map quantitative trait loci in cattle, we're going to need lots and lots of polymorphic genes. And that's really how it started. Of course, Jim came to my office and we talked. He was kind, he was gracious. He had, we talked about some collaboration that never really happened, about mapping blood group genes and so on. It never really happened, but it was an eye-opener for me, and it was something that really turned my career around at that time. So let's take a look at that, because that was a pivotal moment in the history of animal genetics in 1986. The field itself was beginning to undergo rapid changes, right? Animal genetics at that time was animal blood groups and biochemical polymorphisms. And the main society dealing with animal genetics at that time was called the European Society for Animal Blood Group Research. And it was founded in 1964. Clyde Stormont, the person who recruited me as a graduate student at UC Davis before he retired, was one of the founders in this group. One of the people who probably hired Leif was one of those founders as well. Jan Rendell there was a small group of people that all kind of passed through UC Davis learning about animal blood groups and biochemical genetics. And in 1972, the name of the society was changed to the International Society for Animal Blood Group Research, and it stayed that way until 1988. But 1988 then was a critical time because the meeting in 1986, where I first met Leif and Michelle, there were a bunch of young whippersnappers there who were using molecular techniques in animal genetics for the first time. And that became sort of a growth industry within the society, such that the next 2 years, there were as many people working in areas of animal molecular genetics as there were really in the blood groups and biochemical genetics. And in 1988, which is Larry's first meeting, and there's a little picture that I think Larry took of me there with Rudy Fries and the late Nate Fetchheimer, who was a cytogeneticist in Zurich, where we spent a few days on our way to Torino for this meeting. Now, it was amazing that Jim showed up because I knew Jim, and in 1988, correct me if I'm wrong, is when Jim had his first heart attack, if you won't mind me sharing that. And I think it was 3 weeks before the meeting, and Jim showed up at the meeting, like 3 weeks after this. I just couldn't believe it. And he thought it was so important to be there at this transitional period. And this is the time for me to say that Jim ended up serving as the president for the International Society for Animal Genetics for the years 2000 to 2006. So he really led the transition of the society into the modern era of molecular genetics and genomics and beyond. So Jim was really dedicated to the service. He also did a lot of other great service thing. I know the meeting of the Texas Genetics Societies here. He served as president of that society and also as vice president of the American Genetics Association from 1996 to 2003. So Jim did great service to the community, but I think no service was more important internationally than his service as the president of the International Society for Animal Genetics. And that was 1988. So the next thing, and this was touched upon by Larry, this was the Banbury Conference. So this was 1989, 1990. This was another critical moment because Jim chaired this conference and he got funding. I don't know how he got the funding, but he pulled together a bunch of people, leaders from USDA and from around the country, and there were several of us in the room. Larry was there, I was there, and this meeting was critical. This is the first time I remember sitting there, and Jim Watson walks in the room, sat down and listened to the conversation for an entire afternoon. It was absolutely pivotal because it brought together for the first time the leaders of the nascent the Human Genome Project, which also started in 1990. So people like Ray White were at this meeting and others from the human genetics field. Michelle, I think you were there, yeah. And this was a very pivotal period. And as Larry mentioned, this meeting was followed up in the same year by the Allerton Conference. And these two meetings together led to the organization of the USDA National Animal Research Program. This is what we said, yes, we can do this, we can make maps. We weren't talking about sequencing yet. We said, we need the maps, and we need maps spaced by certain markers, and we need polymorphic markers so that we can do what we call marker-assisted selection. It was before people talked about genomic selection. It was really, that was the meeting, and we convinced at USDA that this needed to be done, and that's what led to that program. And Jim served as the cattle coordinator of that program for 15 years, from 1993, from its inception, to 2008. And so those meetings, and then there was another meeting that year, which was held in Edinburgh, the World Animal, what is it, WGALP, World Congress for Genetics as Applied to Livestock Production. And Morris Solar chaired a session there, which also brought together all the people who were thinking about doing marker-assisted selection. These three meetings were a real turning point when we realized that we had the potential to make the maps that would change the way animal breeding would be done. None of our friends in quantitative genetics believed that we'd ever get enough markers to do it. It was all these fights in the bathroom that I had with Dan Gianola and people like that who said, you'll never have enough markers. But all of us believed that it would be done. Michelle really helped us and led the way with VNTRs, the first really highly polymorphic markers in the cattle genome. And we were off, and we were moving. So the following years were a lot of fun. There was a lot of excitement. This is a picture from the 1992 meeting in Interlaken. Oh, sorry guys. This was the meeting from the 1992, picture from the 1992 meeting in Interlaken. And every 2 years we would have this meeting, and the evening we'd have a contest where the people from the different countries would get up and put on a show. You know, the Czechs would get up and sing in six-part harmonies, and the French would do, and the Spaniards would get up in their castanets. Well, we didn't know what the heck to do. It turned out to be, that was the summer of the Summer Olympics in Barcelona. 1992 said, well, why don't we just be the dream team? Well, if I said basketball, Jim was all in on that, right? He was all in. There he is down there at that. That's him at the tie. You can't see it very well. And there's that guy in the front there who's dribbling a plastic basketball between his legs. That's how pathetic we were. But we had a lot of spirit, and we won. We won. And Jim wouldn't have it any other way. We had to win if we were going to go into this thing. You see how serious he is? This was like no messing around. We're in this thing to win. We did a Harlem Globetrotters kind of thing, passing the ball around, and Jim was throwing the ball behind his back and doing all this stuff, and it was great. And that's Sue Lamont there, I think Chris Tuggle, I'm not sure who that, and there's Max in the back there. See Max? There he is, poking his head through there. And it was great, and these were wonderful times. We were making a lot of progress. This is Jim and I at the top of a mountain in Interlaken. Jim didn't take off his conference badge, but which he didn't do often. And John Beaver, who was actually one of my first graduate students, Larry mentioned him. And we had some wonderful times, and I'll skip the story about Michelle George at that meeting, but he had some wonderful times too. But that was 1992, and 1993 was another really important year, because Jim had been mapping genes intensively and collaborating with Rudy Fries, who was really one of the first, if not the first in the community to be doing fluorescence in situ hybridization for physical assignment of genes to chromosomes. So Jim had all these linkage groups that he had defined by somatic cell mapping, and Rudy and Andre and Jim together collaborated to create this first real edition of an anchored bovine gene map. The map had like 350 loci assigned to 19 out of 30 chromosomes, so they were doing a little better, but there was still a lot of territory to cover. 248 of those markers were genes. And I think if you look back in the history, this paper in 1993 was probably Jim's most important scientific achievement. It took them years, to do this. I said, 84 was when the first maps came. So it was really over this period of 10 years that Jim and all the people who were here in his lab were working their tails off to map as many genes as they possibly could with clones, cloned fragments of genes that they were getting from the human or mouse genome. And it was hard work, very hard work. But they did it, and I think this really set the stage. This is the Oxford grid. You can see all the relationships there between human chromosomes and cattle chromosomes. This was the first really detailed Oxford grid, even though it had just 2/3 of the chromosomes. But this really set the stage, and I firmly believe that it was this contribution in 1993 that resulted in Jim's election to the National Academy in 1999, and also led to him winning the Wolf Prize in agriculture. This is really seminal, important, foundational map that changed the entire field. I mean, it led people like Michelle and others to find genes in the bovine genome and know where they might be on what chromosomes. It was really important. And many people, Dave Threadgill and Al Dietz, Ann Ryan, Holly Niebergs, all these people were really involved in these fantastic studies. Laylee, who's also here, I know was involved in these studies. Okay, and I don't want to leave this out because this was also very important. This was also 1993, and with his friend Steve O'Brien and also Neil Copeland and Nancy Jenkins, who are mouse geneticists, over some weekend that Steve's going to tell us about, they came up with this strategy called CATS, comparative mapping of anchor loci, where they could select, they proposed a series of, 300 or so genes that if they had the right probes, they can map it across all of the vertebrate genomes and have comparative maps that they can do deep chromosome evolutionary studies of chromosome evolution and evolution of vertebrate genomes. And so that was a brilliant, concept. It didn't work, unfortunately, as Steve will tell you, because it was really hard to get these probes to grow, but it was a great idea. This was a review of sorts, but this was a very important concept because it got people thinking of how this crosstalk, this conversation, as I used in my title, across maps could work to really understand evolution, genome evolution. So this was published as a Nature Reviews article in 1993. And of course, Jim was a critical part here. 1994, and I'm walking you through here. I'm not going to get to 2024. Don't worry, 2023. But I'm going to stop pretty soon. But this was another pivotal year in the field. And there were a lot of important papers that came that year. But this colleague of mine, who was in that picture there, Øystein Lie, who was an old friend who worked on the cattle matrix compatibility complex with me, I proposed to Øystein that we have a conference on comparative mapping. And it hadn't been, nothing like this had happened before. This was comparative gene mapping in terrestrial and aquatic vertebrates. So it was the first time we had a meeting of all people, Gus, you were there, yep, of people working across all vertebrates to see what was known, what could we learn, what could we do. And unfortunately, my favorite picture of Jim and Raby, I don't know if I gave it to you. I could not find it from this meeting. It's Jim and Raby standing in front of the hockey arena in Lillehammer, Jim with his funny flap hat that he had. And it's the two of them standing there. We were freezing in the middle of the night. We walked into the medal ceremony for Tommy Moe, the American downhill skier who won the Olympics that year, and we all stopped and sang the national anthem in the middle of it. It was an amazing thing to just walk into that. But a lot of people were there. There's the late Glenn Evans, who Richard will certainly recognize, who was a person involved in the early days of the Human Genome Project, Rex Gaskins. And on the bottom there, Michelle, you'll see Moshe Solar and his wife, Mimi. And Michelle, I'm sure that's the back of your head. I'm positive that's you. And it was an incredible meeting, and the post-conference activities were amazing. And a lot came out of that meeting. We even had a yeast geneticist there, Bernard Dujon, who was telling us all this fantastic, we were able to see synthetic relationships all the way from yeast to humans. And just to give you an idea, I'm not going to go through this. Since I found this before I threw all my junk out from my office, I kept this because this program is really historic. The people who were at this meeting and the talks that were given, Steve was there. Look at Don Marzo, people you probably never heard of, but Raby will remember, worked on fish genetics. His title of his talk was Reconstructing the Gene Map of the Vertebrate Ancestor in 1994. There was Jim, there was Glenn, Larry, Norm Arnheim, who I did a sabbatical with, who developed was the person who really invented PCR with Kerry Mullis. He got it to work, Norm got it to work, and Norm developed these single sperm typing methods, which I learned on sabbatical. Other people, Ben Coop, Lloyd Smith, Alan Hilliard from the Jackson Labs, Tom Caskey was there. Morris Solar, Oystein Lee, Lena Peltonen, an amazing scientist who told us about these incredible studies she was doing in Finland in human genetics, Michelle, Leif, Alan Archibald, and others. And it was really, scientifically, it was a terrific meeting. And that year, as I said, 94 was a pivotal year. And I had to refresh my memory on a lot of this stuff because it started to happen so fast. First, there were 2 linkage maps of the cattle genome project. In early 94, one was the Australia map, the Bill Behrens map, which was called, and the other came from Clay Center. I know Steve Kappas was invited here, but he couldn't make it from the USDA. Right? Steve was supposed to come, but he was unable to come. But this was an amazing competition at the time between these two groups. And they both finished at about the same time and made some pretty good maps in the order of maybe 200 to 300 markers on a linkage map, which was pretty darn good at the time. And soon after that, the next year, Michel published a linkage map with a lot of his own markers. And at the same time he did the linkage map, he also did the first QTL scan in the U.S. dairy, in the dairy bull population. And that was really a landmark paper. And that whole period, 94, 96, we published a male linkage map with half-sib families, also did a genome scan, found some more QTL. And these studies led to the first mapping of quantitative trait loci and identification of genes like DGAT and APAF1 and several others that led to cloning, identification of mutations, and elimination of deleterious genes, as well as selection for economically important traits in dairy cattle. So this period, 94 to 96, was really an important period. And here in 1994, Jim and myself at the Mendeleum in Brno. You'll remember this, Ray, because you were there also. So this meeting was in Prague, and it turned out to be a very, very important year, and I'll tell you why in a minute, for cattle gene mapping. And the meeting was kind of a horrible thing because it was held at the former Stasi, like the detention center where the Czech secret police used to do their whatever they would do in there. And it was kind of a nightmare, but it was a great meeting. And at that meeting, I mentioned earlier that I did a sabbatical with Norm Arnheim. And in the lab was a German postdoc named Karin Schmidt. And when Karin was doing a postdoc with Peter Goodfellow, and Peter Goodfellow was at that time dramatically expanding the human gene map by mapping ESTs and other markers onto an RH panel. So you had David Cox mapping at Stanford, and you had Peter Goodfellow doing this in Europe. And Karin was a postdoc in that lab, and she was responsible for making cell lines and for genotyping on the panel. And at that meeting, Jim had been doing, somatic cell hybrids, but he knew about the radiation hybrid work, and I think there will be people here who can give more details about this. But Jim couldn't get the cells, he couldn't get it to work. And we were walking on the streets in Prague, and Karen and I, we ran into Raby and Jim on the street. It's another photograph that I can't find, but I remember taking a picture of you and Jim on the street. And I introduced Karin to Jim, and I said, Karin works in Peter Goodfellow's lab, and she's making radiation hybrid cells. Why don't you maybe you can help Jim with this problem? Problem solved within 2 months. And Jim was off making a radiation hybrid panel. So that was a very important and pivotal moment in Jim's career and in my career. And it was also the meeting that Larry told you about, where Larry had to get up there and sing the fight song. So this is probably Jim's most embarrassing moment. After he had to sing the fight song, Larry had to come down and pay his respects. And as you can see, you had one pig geneticist replacing the other lover over there, Max, being a little jealous. But yeah, Larry, that's my most, this is my most risque photo of the night. But this is perhaps Jim's most embarrassing moment, I think, is being smooched by Larry at the meeting. But we were having a good time. We were mapping lots of genes, and it was just the greatest period you could possibly imagine. And so there it was. The next year, Jim had his panel. He was mapping genes. He did the first example there of cattle chromosome 19. He showed that he can separate two markers on the map that could not be separated by linkage, but he could separate them with the 5,000 rad radiation hybrid panel. And he used one of our microsatellites there, Urbana 59 is one of the markers, and this was how he demonstrated that the panel would work. And then soon after that, a map published with Yaping Yang, and where these very now familiar figures that were dominant for a few years, where he did the parallel radiation hybrid mapping between human and cattle. There was no sequence. So you were comparing maps, and he was comparing radiation hybrid maps. This is quite obvious, this one, because, whoops, yeah, I did it again. Here it is, yeah, give me the signal when I mess it up. But yeah, quite obvious here. Here's the linkage map, here's the paper, here's a big block of synteny, and these are two areas of inversions. And you can see this beautifully in the radiation hybrid mapping data that Jim produced. And so this was a game changer, because up until that time, Jim was doing synteny mapping, and those are not ordered genes. They were just placing the genes on the chromosome. Now, with the radiation hybrid map, you could determine the order of the genes on the chromosome, and this was a really, really big deal. And I convinced Jim at that time to send me a little bit of the panel, because we were working on cattle chromosome 23, and Mark Band was a new postdoc in the lab, and we put a bunch of markers on the panel, and we were able to show for the first time an inversion on the cattle chromosome that has the MHC of the class 2 genes. Completely flipped a big inversion, pushing class 2 genes and as well as other parts of the major compatibility complex toward the centromere of the chromosome. Again, demonstrating the great panel of the resource that Jim produced. Now remember, Jim didn't have to share this panel with everybody. This is an example of Jim's kindness and his generosity, and his willing to share his most precious and important reagent with other people to facilitate their work. And then I just want to give you a little bit of something from my lab, because it's really what led to our greatest collaboration, the collaboration between Jim and myself. It was very important. So I got interested in sequencing ESTs because I was starting out, in functional genomics, trying to make our first microarray. So we did some comparative sequencing of a cDNA library from a cattle ovary that we had obtained, and we managed to get 82 tags. And from those 82 tags, we developed a strategy that ended up really changing the way comparative mapping was done. We simply took those ends of the cDNAs, about 400 bases or so, blasted those against the human genome sequence, found the putative orthologs in GenBank, and then used the comparative mapping information that we already had between the cattle and the human genome to predict what chromosome they would be on, either in cattle and human. And in this paper, the first one there, we actually mapped, we didn't use Jim's panel, we used the French panel, and mapped them in cattle and showed that all the ESTs we were able to predict where they were based on their location in the human map. We could predict the correct location in the cow map. That was critical. And then we did a bunch of libraries, and we did this big prediction of more than 40,000 ESTs, of which we could find 21,000 that had GB4, that was the human genome map at the time, that had that mapping data. And we could predict in the cattle genome where 21,000 genes were, without mapping. It was all in silico mapping, and we called this COMPASS, comparative mapping by annotation and sequence similarity. So I got very excited by this, and I called Jim, and I said, Jim, we can map a lot of genes this way, and we can make a comparative map that would be unparalleled. And Jim was kind enough to send me enough DNA from the cell lines to map a lot of genes. And this is just a progression of the map. The one on the left there's a linkage map in 1996 with a couple of markers. The first map was about 700, almost 800 markers there, the 2000 map, which we then took all these predicted ESTs and we selected them so that they would be on all the different cattle linkage. We had thousands of them, but we selected 700. We got a grant to do this. And so all of a sudden we went from 300 genes on a synteny map to more than 700 genes where their order was mapped in the cattle genome. And this was a full-page pullout at that time in genome research that I found plastered on the walls of many laboratories as we went around the world. And again, this was something that was totally enabled by Jim's generosity. A few years later, graduate student in the lab, we made the next map, which was 1,400 markers. And then another big breakthrough was we had back libraries, and we were able to sequence the ends of back clones using the same COMPASS markers, and we were able to select markers approximately every 1 to 2 centimorgans in the human genome and map those onto the cattle genome. So this map, the one that you see with Dennis's picture in 2005, was the resource map that was eventually used by Richard and the folks who sequenced the cattle genome. This was the scaffold for the bovine genome sequence. We didn't have long reads in those days, and we had to use maps, and there was lots of maps around, and Richard and George in the end, after, it's another thing a book should be written about, I think, but in the end they said we got to go with the map that makes the most sense, and they used this map. So Jim, contributed again a resource that allowed the cattle genome to come together into ordered sets of genes and markers on the cattle chromosomes. And that really changed the direction of my career. I totally got sucked into the O'Brien-Womack thing of comparative mapping. And with these very highly detailed maps, we can produce for the first time highly detailed comparative maps of the mouse and the cow and the human, right? These are phylogenetically very disparate, and so they formed a really good basis for understanding chromosome evolution. Just for one chromosome, we got the cover of Genome Research. That was what you could get the cover of Genome Research for in 2003. But there's lots in this paper, and one of the things was it began to dispel the myth that chromosomes broke randomly in evolution, sort of the Nadeau hypothesis of random chromosome breakage in evolution. We actually saw areas of shared breakpoints, which later on with Bill, we showed these were reuse breakpoints in evolution in independent lineages, sites of chromosome instability that broke and shuffled over and over again, even in different lineages. So this had implications, many implications for cancer. I'll talk about that in a minute. But this kind of blew the field open, if you will, for detailed comparative mapping, and it was the livestock species that provided the outgroup, really, the Laurasiatherians, cattle versus the Uarconta, where mouse and human were residing, the primates and the murids. Okay, and then again, I think this 3,000 marker map was extremely important. We mapped 3,000 back-end sequences. We had 3,400 markers. 3,200 of them had sites in the human genome. So we call these casts, Steve. That's why, because they're comparatively anchored sequence tagged sites, okay? Because they were back ends. And so we didn't know if they were genes or not, but they worked absolutely beautiful as comparatively anchored markers. Had 90% coverage against the human genome. We found 7 new blocks of synteny and extended 79. And this really opened the door to the next phase. And this is when Jim, visited the lab during this time. That's Annalee Everett Vanderwin, a student of mine, Dennis, who worked 9 years in my lab, at first as a postdoc, then a visiting professor, and now a full professor in the Royal Veterinary College in London, where I know you were, Dean August. Sorry, Jim Womack. I'm sorry, it said light in here, but it's Jim, Annalee, Dennis, and myself. And it was faded. It's faded because it hung on my wall in my office. I love this picture, and it got a lot of sunlight, and I don't know who took it, but it was there on my wall till I left the University of Illinois in 2011. But this, I think, was our crowning achievement, and would not have been possible without Jim's guidance, Jim's generosity, and his willingness to say, okay, you do it, which he did, and we did, and we did it together, and it was fabulous. Now, enter Bill Murphy and Steve O'Brien, because I called Jim, I said, this is a great opportunity to do multi-species comparative mapping. Nothing had really been done. We had a few good gene maps, the pig maps came along, so I called Larry. I got a hold of the horse map people. I know that the cat map would be available, hopefully through Steve. And we had just the human and the mouse sequence at that time, some rat sequence, but we still used the rat map, and we had then just maps. And we had to figure out how to integrate sequences and maps. That was not trivial. But with Bill and with I skipped a little bit of that story. Let me just check the time because I see that people are falling asleep here. Yeah, because I'm almost done. So I called Jim. I says, okay to call Steve, and should we invite Steve into this collaboration? He said, sure. And I called Steve, and he said, I know just a guy to work with you on this, and it was Bill, who was doing a postdoc in Steve's lab. And together we did this wonderful study where we did a very detailed reconstruction of the Boreoeutherian ancestral genome, and all those colored blocks are the areas, ancestral blocks that we could compute using the MGR algorithm, which was developed by Glenn Tesler and Pavel Pevsner, who are also collaborators on this paper. And this ancestral reconstruction covered 50% of the human genome, but it was pretty good at that time, considering we only had maps. And so that period was critical. I'll go back just 2 years and say this was another important document that I found. This was a National Research Council report, and this is the group. I don't know if you were there, Richard, at this one, but this is the meeting where Steve and myself and Larry, we kind of fought it out over which genome would be the next genome NIH would fund to be sequenced. And everybody had their favorite genome. Steve had the cats, and Larry and Max were arguing for the pig, and we were arguing for the cow, and and a few people argued for chickens. Matt, I read the thing, and even Max argued for chickens, which was very selfless of him to do at the time. And Richard, you were certainly there at this meeting, and I hope you'll talk about this. I hope you will talk about this tomorrow in your. George was there. Okay, thought you were there, but I hope you'll talk about this because the whole thing. I'm going to leave it out. Larry talk about it. It's a long story, but in the end, Jim's leadership with the governor of the state of Texas and his connections to the people in Texas and Baylor, and really led. It was really in the end money that helped decide, because this was a $50 million genome. It would originally, at the time we had this meeting, there were $100 million genomes. And Eric Green was arguing, just do targeted sequencing and you'll find everything. Read it. I read the report again. It's really illuminating to go back and look at people's arguments. But Jim came forward and took the leadership and nailed it. And we were on our way to sequence the cattle genome. And there it was a few years later. Thank you, Richard, for your leadership and all the work that you and George did to bring this about. We had our first draft of the cattle genome sequence, and this is where I think we end really for Jim. Anyway, sorry? Yeah, and as some famous Texans have said, Willie Nelson, the road now goes on forever. Genomes, we just keep going. Larry mentioned the Earth Biogenome Project. We're trying to sequence, 1.8 million eukaryotic species. Richard was also at that initial meeting we had at the Smithsonian in 2015, where we said, is this a crazy idea? And they said, not so crazy. And we spent 5 years developing the concept for this, or 3 years, and eventually published a manifesto in PNAS, and we're 3,500 genomes into it, which is pretty darn good. And I said the road goes on. Jim was very proud of this. He wasn't on this paper, but this was the paper where we were able to do a really detailed reconstruction, a postdoc of mine, of the mammalian genome. I shared this paper with him in 2022, and he was as proud of it as if he was an author on this paper. We were able to do a detailed reconstruction of many different ancestors and extending the Boreotherian, which was 50% in 2005 to like 95% coverage against the human genome. And all the way through these ancestors leading to the human genome that you can see. We could determine the number of chromosomes the ancestor likely had. And dating back to Jim's first love, what are the ancestral blocks? We had 2,557 synthetic segments shared between all of these mammal genomes on an average of 880 kilobase. And something magic about 1 megabase in mammalian genomes. And we're still trying to understand that. But these are the building blocks of all the mammal genomes we see today. They're just shuffling of these 2,500 segments in different orders and combinations. All extant mammalian genomes are comprised of these fundamental building blocks. And Jim would be amazed to see this. This is unpublished data, but thank you, Fiona, for coming. This is a new collaboration with the BAT1K group, Maxim Prolutsky, a postdoc from the Ukraine, Dennis Larkin in Dennis Larkin's group, Michael Hiller in Frankfurt, and Emma Teeling in the entire BAT1K. Just think of this, folks. This is 103 reference quality genomes in one mammalian order. That's science fiction. That is just hard to believe. These are genomes that are 100 times better than what the 2003 genomes. These are chromosome scale assemblies. They're not T to T, but they're close. 103 in the same order. And this is one representative of each of the BAT orders. Can't show all 103. And we did this first reconstruction of the chiropteran ancestor, the ancestor of all bats, using 103 genomes. Just something that's hard to believe. So over the years, we had so many good times. I'm sorry to run over, but I'm very enthusiastic about all this, and I won't take away from much more from your bar time. But this was a great honor to have Jim and Raby come to our dinners every year at PAG when all the mappers were there. Mark Band's in that picture, Annalise at the end. I know you probably can't see all of these. Here's the picture, sorry. And these were just wonderful times, and Jim and Raby joined us for many years at PAG to celebrate all the accomplishments that we had achieved together. And so I would like to close with this wonderful picture that I love, my favorite picture of Jim and Raby taken at Jim's ranch. And I'd like to close with a very personal story. Raby, I hope you won't mind me sharing this story. I'd been trying to reach Jim for a few days when he was sick and learned that he had returned to the hospital. And I'd spoken to Jim and Raby about a week before, and it was clear that Jim's health was rapidly declining. And I tried to reach him, I couldn't reach him on the phone. And then the phone rang, my cell phone, and I saw Jim's name come up on the phone, but I was really afraid to answer the phone. But Raby was on the other end of the phone, and she was at Jim's bedside. And she told me that she had told Jim that I had called and that he wanted to talk to me, but was really unable to do so. But as she was speaking, as I recall that story, she suddenly said, "Wait, I think Jim wants to speak with you." And she couldn't believe it because Jim hadn't spoken a word in more than a day. And the next thing I heard on the other end of the phone was, what do you think? "Harris, how are you doing?" Strong and clear, as if he was in the next room, like nothing was going on. It was incredible. I couldn't believe it. And he told me in a few words, we talked for a couple minutes, how grateful he was for his life and for our friendship. And after a minute or so, we started to say our goodbyes, and he said something that really startled me. He said, "I love you." And I replied to him, "Jim, I love you too." And those were the last words that we exchanged. And in a couple of days later, Jim was gone. I reflected deeply on my last conversation with Jim many times, and the one thing that comes to mind was a Beatles song, a song called "In the End." It's the last track on the famous Beatles album, Abbey Road, that I'm sure that Jim never heard. And the words go like this. It's, "And in the end, the love that you take is equal to the love you make." It's a very deep truth. However, for Jim, it was an exception, because I think that Jim made and gave more love than he took. All of us know this in this room as recipients of his warmth, his generosity, and his love. So thank you, Jim, for the great legacy that you left us as a scientist, a teacher, and as a good and decent human being. Mapping genes has indeed been good for and good to all of us, too. Thanks very much for your patience, folks. Thank you, Larry.
I think rather than taking questions now, I think we'll break.
There are no questions.
And
Yeah.
I'm sure once I get that glass of wine in his hand, he'll be more than glad.
A gin and tonic may be better. Thank you very much. Thanks, everyone, for your patience. Sorry.